Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Ferdinand von Meyenn from the ETH Zürich on his show. Please let me briefly introduce you to our audience. You got your PhD from the ETH in Zürich in 2011. After a short postdoc at the ETH, you moved to the Babraham Institute in the UK to do a postdoc with Wolf Reick. After that, you started your own group at King's College in London in 2017. And since 2019, you are assistant professor at the ETH in Zurich. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Well, I'm actually, my parents are both scientists. And so that's a little bit how I got into that area. My mother is an MD. And obviously I was for a long time very much connected to that. Initially, actually, I started studying biochemistry. So I had much more the link into, a little bit linking it to more the chemical side as well. But very quickly became very excited about the biomedical approaches. And that very much linked them back again for my parents to the side and have been stick with that. And I think I really like this idea of how processes work, in particular at this biological level. So actually how they translate us into humans, what they actually mean for our health. And that actually, if I look through my career, most of the thing I didn't follow a very exact path, but overall always following some biomedical related interest. And I think that's where I'm still today.
So let's talk about the science you did. And I want to start with when you were with Wolf Reick. During this time, you worked, obviously, one could say, on DNA methylation in naive embryonic stem cells and the cause for the global demethylation in those cells. What did you find about the cause of the global demethylation in naive ESCs?
So this was a project we started because we were very interested in how early embryo actually works and what of the dynamics are happening in there. And so we used the naive stem cells, which at the time also by Austin Smith and others have been initially described, and they looked into that. And we tried to better understand what are the methylation pathways. And we were lucky that we had actually access to many different knockout cell lines, which remove the key enzymes which are related to DNA methylation and maintenance. So both the de novo methylases, the DNMT3s, the maintenance methylation system, but also the TET enzymes. We had the triple TET knockout cell lines. And in addition to that, also, we started looking a bit more into those enzymes which are kind of cofactors like the UHF1. And the finding we had at the time was that obviously, and this was work done also before, that there is a global methylation difference between the naive and the primed cells, the stem cells. And if you now looked into transition from prime to naive using this 2I live cocktail, which people have been using, basically allows cells to transition into that phase, and doing that in the context of the various knockout cell lines, here we could find that removing the DNMT3s wasn't actually making much of a difference. As you would expect, equally the TETs were not that essential for the process, but particularly removing DNMT1 or UHF1, these seem to be the key drivers of this demethylation process. And then maybe more by an accident, I could say, at least initially it was a bit of an accident, we looked at the regulation of UHF1. We generated a cell line which coupled UHF1 to a GFP. And we actually want to look at the UHF1 localization. But what we found is that actually UHF1 levels suddenly dropped dramatically. And so this kind of gave us an insight that there was some regulation. And then we started looking more at the protein level, at the RNA level, and actually found that this seemed to be the key regulatory part in the whole process. So removing UHF1, that way we disentangled the maintenance methylation system. We basically do not maintain methylation. And there we then also found with the other regulation part that how UHF1 is brought to the replication fork. Put that all together at the end, we could really put it together. And I think we have somewhat of a mechanism that now the loss of UHF1 together with loss of the epigenetic modification K9 dimethylation, which normally also find in association with DNA methylation, these two together seem to be responsible that suddenly we do not maintain DNA methylation in that process, and very rapidly methylation is lost.
So how is... how is it lost? Is it like no longer expressed? Is it degraded? Is it both? Or at which level?
So we find to a large degree that the protein also there must be some degradation in there. So it's been removed. We also find that there's a transcriptional regulation in there. So really at the RNA level, we find that too. And interestingly, it's to a large extent actually at the UHF1 level. So the DNMT1 levels seem to be less affected. And I think that was for us the exciting part, that really here it's the recruitment part which is key and less the enzymatic activity part which might be impaired. And so looking at that, we find that this is in our system the case, but when we then also look at various other in vivo systems, also in the germ cells, we believe that in all of these systems, this seems to be a common motif. And so at the end, the level of maintenance by the level of how much UHF1 is there, that might be a way to describe how quickly systems can demethylate.
So this is then a function of dilution? Because in a replication, you just don't put it back, or is it also like because it's actively removed and then not put back?
So our data indicated to a large extent this is actually a dilution process by not putting it there. If we now look at newer data also from other labs which have been generated, there clearly seems to be also a contribution of the TET enzymes. Our model actually indicated that this is very minor, but that could also be obviously at the various phases. And also then if you look at, for example, the germ cells, there probably the TET enzymes play even a bigger role. And equally, in the very early transition from the zygotic stage to the ICM, also there's work from Petra Hašková and others, which suggests that indeed also TET enzymes seem to be playing a key role in that process.
Yeah,
but to model, we find that actually a large part we can really model just by passive dilution.
Mm-hmm. But if the TET enzymes would play a role in this, then obviously the downstream modifications would also be interesting to look at, right? Because they might have other regulatory roles.
Exactly. I mean, definitely hydroxymethylation seems to attract and bind to other proteins. And so here, the functionality might be also quite important. At the same time, one thinks about the loss of DNA methylation, the activity of the TET enzymes, hydroxymethylating the methylation site, also has a similar effect because at that point, maintenance doesn't work anymore. And so the net outcome of hydroxymethylation might be not too different from actually not maintaining it. And so I think these two could even, at least in a replication-dependent manner, work together in a very similar fashion.
Next, you recapitulated primordial germ cell specification in vitro from naive embryonic stem cells and characterized the early events of epigenetic reprogramming during the formation of the human and mouse germline. How did you transition into this, and what did you find there?
This was a bit a process because there was the whole interest in how this whole process might be regulated. Again, thinking about the DNA methylation dynamics that we find in germ cells, obviously a very exciting part because once they're fully kind of after the primordial germ cell formation, we know from in vivo data that these are really the lowest methylated system. So really here, a large wave of erasure of epigenetic information happened. At the same time, studying germ cells is always a bit tricky because they're very scarce. There's not many. The human system is very limited, also the accessibility, because obviously this falls in the period where pregnancy is not yet detected. Getting human samples in all cases very challenging. And so there was really the interest to generate those in vitro and work with them. In parallel to that, there was the work from Hayashi, which had been published recently, which really showed the path towards germ cell formation. And so on the one hand, we started using their protocol for the mouse system and tried to recapitulate, reinstate that. And then while they initially had primarily looked at the transcriptional and the formation of these germ cells, so the identity, and actually in parallel to us also did the whole work where they looked at the epigenetic changes in these germ cells, we also were interested in the human system. And looking into the human system was a bit primed by work by Austin Smith again and others, which had shown that also human cells can be put into this naive state. And so then comparing the mouse germ cell specification path and what Hayashi and colleagues had done, we were wondering if this can also be now translated to the human side. And so we took a very similar approach where we went from these naive cells, we capacitated or primed them towards a state which we believed was similar to the point where then also the germ cells get specified, and then went into a similar process where we activated various factors, added the growth factors that normally are known also in vivo to promote germ cell formation. And it took quite a while to play around and actually get a protocol that worked. I believe by now people have optimized it, and even various other iterations have been found that might even be a bit more successful or a bit easier to follow. But at least for us, it was quite interesting to see that the dynamics seem to be slightly different between human and mouse. Again, something which may not be too surprising, that the human dynamics of development are different to the mouse. But it was nice to see that also this holds true in the in vitro system. So here really it's the factor, there must be somewhat a cell intrinsic factor which also dictates a bit the pace at which these epigenetic changes and also developmental changes are happening. And what we found is that indeed we obviously have this remethylation phase from the ICM. So the cells first a high expression of de novo methylases, they gain methylation, and then we have a state where probably these germ cells are formed, or these primordial germ cells are formed, and then they go through a wave of demethylation. And in the human system, we're not able to push them that far, just because then the system becomes very big. So we use an embryo body type system, they become too big, so there is a limitation on timing. But we found initial signs of demethylation, and equally in the mouse system, we find very nice demethylation paths. And I think that now also by others have shown that this fits in the overall hypothesis, and also followed up, and much more markers have been looked in there.
So after that, you returned to the ETH and set up your own lab. I'm always interested, and maybe others are as well, how this transition happens, because you have been working on something and you transition to your own lab and are independent now, and then you maybe can take the project with you, or you'll go into a completely different direction. How was it for you, and what did you want to achieve or do research on in your own lab?
So maybe it's that I'll have to do a bit of a jump back even into my PhD. So I started off my PhD already at ETH, and here was working in the lab of Markus Stoffel, working on endocrinology, metabolism, in particular obesity. And so when I joined Wolf Reick, after finishing my PhD, which was all about pancreatic, islets, a bit of metabolism, really focused on that area, not at all touching epigenetics, I really had kind of the aim to kind of re-explore what I could do. And so the epigenetics landscape at the time sounded fascinating. I had actually very little idea of epigenetics at the time. And so I joined Wolf Reich's lab, and I think it was a fantastic experience, and really I learned a lot, and very quickly. I mean, it was a fantastic environment there. But already then, when I wrote my fellowship applications, I put in there that the overall goal of my scientific career somewhat would be to bring these two fields together. So start thinking about metabolic changes and how metabolism and epigenome and epigenetic regulation may come together. And while I think initially this was a fellowship that I wrote and felt, oh, this sounds like future interesting, I think the closer I came to starting up my own lab and thinking about these things, this made more and more sense. And so this actually then, when I also started applying for positions, I was really looking for this space of interest from the institutional side of person working on metabolism, nutrition, metabolic health, but in the context of epigenetics. And here at ETH Zürich, there was at the time an interest in finding somebody to take on, particularly with also the link to nutrition, to think about how does long-term changes, how does nutrition affect our health, metabolic health. And I think they like the idea that maybe bring in the epigenetic angle. And so with that, I approached here ETH, I applied for the position, and somehow they seem to be liking the idea. And since then, I've been here. That being said, the whole journey actually of independence started even a bit earlier. So initially, while I was starting or thinking and shaping what I want to do, I first joined for roughly a year King's College in London, and there the Department for Genetics. And they already started shaping a bit the area. That was at this time a bit more still focused on how stem cells, pluripotent stem cells would be shaped by metabolic changes, and how we could actually use that and maybe also better understand developmental processes through metabolic regulation affecting the epigenome. And this is actually, since we have been here, a little bit the theme of trying to understand how are metabolic, called environmental effects impacting on the epigenome, and that in various systems. So we are overall working rather tissue agnostic, I would say it. Also, we have obviously an interest in metabolic tissues, but from the project we do, they all cover areas from in the liver. We are a bit interested. We are interested in the adipose tissue recently quite a bit, but also in stem cells and stem cell dynamics and how stem cells are affected by metabolic cues, which then could change the epigenetic effects.
Yeah, I guess we will go into a little bit more details on this, what you did in your own lab. But if the first thing you did, or at least that was published, goes exactly in that direction, right? Metabolics and epigenetics, because you looked at histone lactylation. And I guess to lactylate histones, this already plays into the kind of theme. So what did you find about this histone PTM?
Yeah, so this was a project which was inspired a bit by a recent publication, which was very early when I joined here. So the work also which showed for the first time that actually histone lactylation exists and suggested that it might play a role in macrophage biology. And we at the time basically started hypothesizing, so if we have lactylation, I mean, it makes sense that we may find that because there's a lot of lactate, there's lactyl-CoA, so this could work. But it would also really give a link from metabolism to the epigenome, so really instructing the cells. And so then we basically decided, let's look for that. Where is it? How is it localized? At the time, we barely knew anything about it. The tools were not really there. And so we very quickly then got the antibodies. We started doing all of the ChIP-seq profiles. So actually, we did mostly the cut and tag. So using a methodology slightly, I think, a bit more advanced or now quite well-established protocol. And we found actually that lactylation is pretty much found in many cell types. So it's not like a very unique one that you do not find at all. But the localization is still a bit more specific. Overall, it follows the tracks of histone acetylation. So we find that actually K27 acetylation and K18 lactylation are overall similar. Yet the lactylation seem to be even more specific for enhancers. And so when we looked at it, we did it for many different tissue types and cell types and also slightly different stages, we actually found that this was a very good modification to tell us which enhancers are active in their respective cell line or tissue type. We at this point have not yet fully understood why that's the case. We believe that P300 is probably also the writer of that modification. Also that probably also others could do it, and this is very challenging to really test that. But it seems to be linking a bit to the metabolic side. Yet what we do not find is now that if we feed, for example, and I don't think we showed that in our manuscript, but we did the experiment where we fed mice with high levels of lactate. We played around with these things. These did not directly now affect the lactylation levels. So this direct link from nutrition to epigenetic modifications in the muscle, for example, that we couldn't show at the time. But we clearly found that the lactate levels in the cell culture, these could really be influencing histone lactylation levels. So there seemed to be a direct link, but at a systemic level, that's still a bit less well described.
So there are like...
We at the moment...
There are like three keywords. Maybe you will come to that, what you're just saying, but I'm just throwing it out now. So there's the reversibility, the abundance, and the sterics. Did you look at any of those, and what can you tell us about that?
So it's part of things we're still doing to a large degree. That the challenge here is how can we actively place histone lactylation at one side to actually test functionality, and equally binding. So there's many of these factors which turn out to be a bit more challenging. What we have now started doing is using this histone code expansion. So basically that introducing specific modification, specific sites to test that. And that is basically work where we also got help from Simon Elsässer and basically also some tools from them. So where we now can engineer histones, particularly the histone 3, which then contains an either acetylation or lactylation at the respective site, and start testing that. And initially, we just basically validated the antibodies. We can show that they are very specific. So that was retrospectively a very good information to know that indeed our antibodies also pulled out the information we wanted. And now we're starting looking, and we don't yet have the final answer to that, whether actually lactylation binds other proteins and basically other kind of cofactors, and that might explain it. We have some preliminary data which indicates that lactylation and acetylation have slightly different dynamics, how quickly they're removed and replaced. And so that might be a functional role in there. But this is still ongoing work, and it turns out because it's rather low abundance, so the levels of lactylation over acetylation are magnitudes lower. So that's a bit more the question, what is happening there. So it's still a process we are involved in. I don't think lactylation at the end is going to be the modification which explains everything. I mean, that's probably was not expectation. There seemed to be a link to the abundance of lactate in there. And so that experiment we did, where basically we just add a lot of lactate to the media or lactic acid, and that clearly has an effect on lactylation. But really the biological function, I think this is still something we need to explore. And I believe that maybe in this dynamics, that might be a key to it, that how long lactylation lives seems to be different to how long acetylation lives. And so maybe a site marked by lactylation may be then a bit longer there maintained, and so may have a different effect on the dynamic dimension, how strong then the expression of the respective gene is.
Since I'm an avid runner, it might be also linked to physical activity if you accumulate lactate in your muscle, maybe, because then you don't have the active transporter from the gut to the blood, but it's there already, right?
Yeah. Also, we actually did these experiments too. We put mice on the running wheels.
There you go.
Quite significant. So we played around with these things. At the end, we have some minor effects. They're not yet so conclusive that we would that I could now say this is clearly lactylation is now an epigenetic memory of a runner versus none. This obviously was the initial when we started this whole. The hypothesis was exercise would promote high levels of lactylation, and we may be able to mimic that by nutrition. And I said this didn't work out the way we initially expected it. We rather found where it's localized. But it's still something we are following up. And maybe even if we the main goal now we decided is can we show what factors are bound, and can we show that adding lactylation to a specific gene by basically this engineering process, can we then drive gene expression in a different manner than by just putting acetylation on there? And I think if that's the case, and these experiments we are currently doing, this really been exciting to really then test in more detail. Are there even small changes in that abundance, which really then could also have a biological outcome?
Then moving on, you found that capacitated HPSCs are transcriptionally closer to post-implantation epiblast than conventional HPSCs, which prompted you to comprehensively study epigenetic and related transcriptional changes during capacitation. This is now mainly from your abstract, so I didn't know what capacitation was. So maybe you can briefly talk and explain that process and what you found about it then in the epigenetic space.
Yeah. So this is a bit of a work following on from the work I did with Wolf Reick at the time in collaboration with Austin Smith. And so here we again looked at the effects in human pluripotent stem cells or embryonic stem cells on the epigenetic changes that happen while these cells transition from this naive to the primed state. And this whole work was a little bit inspired by what we found, that if you have long-term human embryonic stem cell culture, they accumulate some epigenetic aberrations. Maybe differentiation effects are different. So there's also now more recent work showing that embryonic stem cells can slightly differentiate based on their epigenetic profile. So they may lose certain potentials for certain lineages. And so the idea was now, since this naive state seems to be more or less removing most of the epigenetic information or putting them back into a very much earlier state, could we now use that early stage and pushing cells into that stage to really make them fully competent for all lineages later, or removing any of these biases? And so kind of that was the initial hypothesis we started without. And then we start looking, going from cells which were derived in this naive condition, or cells which were derived in this primed condition, and then reset how similar they are and how much actually now going back from this naive state to a primed or capacitated stage, because capacitation then also, this comes from the idea, the naive cells are very poor in differentiating into the germ layers, whereas primed cells are much better. And through going through a so-called capacitation stage, we can actually make these naive cells again fully competent for germ layer formation. And so we looked at the epigenetic dynamics in that process, And what we found, that this works. We gain many of these epigenetic modifications are regained and go back to the initial stage. In particular, we found at the transcriptional level that while we go there, we again this capacitation, we go back close to the epiblast stage. One key thing probably we found in there was the X chromosome. While that is many times in long-term maintained classical embryonic stem cell cultures, there we find this X chromosome erosion. Putting it back in this naive state, we largely find a reactivation of both X chromosomes, and this capacitation seems to be more or less silence only one copy, so we get back to this normal stage where we have one active and one inactive X chromosome. Equally, we found a role of the polycomb, so basically how much PRC2 activity is there. And so in that case, we found that actually PRC2 inhibition does not prevent this capacitation, which a little bit was a surprising finding given the huge importance of polycomb normally in early development. So it seems this early phase was not affected by that. And finally, one thing which probably is on maybe more on the negative side is, but we had seen that before, these early naive cells, they seem not to protect the imprints. And so while normally we have obviously very nice imprint control and we have one methylated and one unmethylated allele, these gets largely lost during this resetting phase of putting in the naive condition. And unfortunately, that also is once lost, not reinstated. But overall, basically, it gives a system where you can now explore that further. And I think other colleagues, also Ryan Lister recently had a nice paper on that, where they looked into all of these various ways of using these more naive-like conditions to remove some existing or maybe deleterious epigenetic changes which may prevent further development. And by putting back to this more naive, or at least pushing more into this naive state, you can remove that and go back into fully competent stem cells.
Moving on again to more work that you did. You also worked on epigenetic clocks, and this has been done in several ways and in several facets. And you the first clock you worked on was based on ATAC-seq, and the second one was called then SC-EpiAge. So what were the advantages of those clocks? Why did you even look at those? And what were you able to do with those?
Yeah. So this maybe it fits a little bit to this whole transition when I also started thinking about my own lab. So the very first clock actually we worked on was still with Wolf in his lab, with Wolf Reick, and Olli Stegler was involved in that. That was, I think, the first mouse methylation clock a few years after Steve Horvath published his initial clock. And that is a work that still was done at the Abraham Institute in Cambridge. And there we basically initially set I would say, the foundation for all of this follow-up work. We really looked at how is aging can be modeled. And I think our work more or less showed that the mice or other species equally have a clock. And I think since then, obviously, this field has gone way beyond of that and really shown that epigenetic age and chronological age are very much linked, and we can really use it as a biological readout. And so when I came here to Zurich, we want to continue that work, I think also with several colleagues. And so the work on the ATAC clock was a work together with Alex Ocampo, who came back from the States. He was working before with Belmonte, and so has done a lot on this reprogramming. So also had quite an interest in epigenetic aging in various ways, how the epigenome changes during the aging process, but also maybe during the rejuvenation phase. And there was some other work also at the time which showed that transcriptional information can also be used to predict age. So really there seemed to be not only the link at this very, I would say, slow-moving modification, DNA methylation, but maybe at this much more fast-paced stages. And so the question was, is now chromatin accessibility maybe a way to more learn about biology? Because I think one key drawback of many of this methylation aging clocks so far is that really the interpretation of where the CPGs are lying that actually contribute to the aging effect, or where we kind of can make the interpretation, which are used to model now aging, and linking them to a biological function has been very challenging. And so the idea was now, if we would look at chromatin accessibility, which might be much tighter linked to expression, maybe this would now reveal new signs of how aging works, what things are really changing. It's not like now we have this really worked in that way exactly, but it was actually quite nice to see that using chromatin accessibility was a very good proxy again to model age. And so this worked quite nicely. This is now done in human fibroblasts and in human samples in the blood. And there again, we nicely see that indeed chromatin changes, despite them being probably more dynamic than what you expect. There's still a lot of change which span a long time period and are useful to predict age. And all of it, the question will be, and we have started discussing that, to maybe even go on the single cell level, to asking the single cell heterogeneity and looking at single cell attack data. Could that also link back to the age, and could we use that? And a bit following back on the work on the methylation clocks, because at the time when I was also leaving Cambridge, all of this work also from other colleagues in Wolf's lab showed that we can look at methylation at single cells and link that even to transcription. And so we asked if, the methylation clocks, maybe the features of the methylation clock can be better understood if we actually look at single cells. So really was the question, is every cell aging, or is the tissue aging? Is the proportion of old and young cells changing? And so we decided, well, let's do that experiment in I would say in a very, not simplistic, but I think we try to be as simplistic as possible. We collected blood from different old mice and did exactly that experiment. We just ask, what's the methylation? Can we just directly predict age? And we initially ran into some issues that obviously having sparse sequencing data for methylation from single cells just was not compatible with any of the clocks developed at the time. Because most of these clocks rely on that you see a defined set of CPGs. And so this was in the work also, particular Mark-Jan Bonder should be named here, who's been a key contributor, and really a lot of the modeling was done by him. Then trying to better understand what is the best way now to do that. And at the end, the approach we took in this single cell age modeling is a reverse step. So rather than look for each CPG, we try to construct based on public and our own bulk sequencing data, how would we imagine the methylome to look at every age time point? And once we had constructed that, we then basically use the single cell data to map it on that and see which is the close resemblance. And having done that, actually it was possible to, again, construct a methylation clock. The key features here are that on one hand, they actually work very nicely also on very sparse data. So they're not only for single cell data, but I think also very useful now, and I hope people will start using them more to predict age of sparse data sets, sparse sequencing data sets. We're actually using it ourselves quite a lot now in that format. And also it showed that single cells seem to be aging. So we actually, if we now look, obviously every individual CPG, every individual cell has a certain error. The error is much higher than you would have in bulk data, but still it's, I think, within the technical range quite good. And I think the most key finding was there that if you look now at the various ages, we have quite a large age span of cells, indicating that indeed there is biologic heterogeneity in biological age at every time point. So it's not like just all cells are the same age and we just detect technical difference. We really detect, or we believe we detect differences in the biological age at every time point, indicating that really the age trajectory is a combination of you have young and old cells. Every cell can age, and I think that kind of now adds a new dimension to a better look into the aging process.
And what do you see when you use the ataxy clock? Do you get more peaks? Do you get more open chromatin in old mice or in old cells? Or what do you see there?
It's actually not too different than what we found with the methylation clocks, that there is both sides gaining and losing accessibility. So it's not like just the chromatin opens up. But I think there's a trend. This is maybe also what we show in the transcription level, that it feels we lose a little bit of control with age. Meaning, if you look at the number, so if you look at the transcription levels, it seems to be a little bit of a, what's the best way to put that? Those genes which are very highly expressed, maybe they lose a bit in expression. Those which are very low gain a bit of expression. So there seems to be a bit of loss of control. I think that's maybe the easiest way to say with the aging process. And in the single cell, the surprising thing is we even see that at those cells which are predicted biologically to be older, that there we have a bit of this effect in there too.
And let's move on, because at the end of last year, which is now 2024, you looked at the connection of obesity and epigenetics, and this was then published in Nature. Can you talk about this study, which seems to be very interesting and very influential potentially?
Yeah, this is, I think, one of the first studies we actually started when I joined here in Zurich. And so we basically were at the time wondering, how is now body weight related to long-term changes? And so there's obviously a long-term observation that people find that gaining weight is a rather easy process, losing weight is a rather challenging process. And I think probably more or less everybody will experience himself or herself or with colleagues and friends around. And so for us, the question was, is this now just so are there any bases on there? Since this also is a long-term process, all of that kind of said maybe there could be an epigenetic addition or some epigenetic role in this whole process. And so what we did at the time, we had luckily access also in contact with clinicians who have been doing many of these studies where people who are very overweight, so people living with obesity, at some point, depending on the body weight and the BMI, it suggested that normal dietary interventions are challenging. And so then people can go for bariatric surgery, in which case the food intake is limited because of the constriction or the restriction, how much they can eat because of it's a physical limitation in the end. And so this is normally a way which is very successful to lose weight. But yet, if we look now at the trajectory of people who did that, we find that many of them still, after an initial very efficient phase of weight loss, they start regaining a bit of weight. And the fantastic part of the studies and these clinicians, they had also sampled adipose tissue, particularly the visceral adipose tissue, but also subcutaneous adipose tissue from those patients during the operation. In particular, one study was quite unique in that this was a two-step surgery process. So people went for surgery, they lost weight, and then after 2 years follow-up, they went for another surgery where the stomach was further a bit remodeled. So this was the second step of this bariatric surgery. And so there was a chance to collect a second piece of the same tissue. And so that gave us a unique opportunity to start now looking what is now happening in this adipose tissue. And we started doing single nucleus RNA sequencing because the adipose tissue is, like most other tissues, quite a heterogeneous tissue. And so doing bulk analysis many times, I think, is very challenging. And the data that one gets out of that is very hard to interpret. And what we found is that we find a clear distinction between people living with obesity and lean individuals. As you might expect, that the adipose tissue really responds and changes in response to this higher nutritional intake, and so adapts to that. The adipose tissue also increases in size. The adipocytes mostly also become larger. And overall, what we find is that the functionality probably in the obese state is less there. So we find also a bit more inflamed state. And yet the overall adipose tissue is not fundamentally changed. But what's then the surprising, or I think very exciting for us, the part is that after weight loss, and if we then again looked at the samples and did the single nucleus RNA sequencing, we find that many genes which were deregulated in the first place remain deregulated, despite that these people experience a significant improvement in their metabolic health. Many metabolic and health parameters have been improved. And so really the surgery, weight loss improved health and was really successful, yet signatures of that prior phase were remaining there. And so that sparked us to look in more detail. And as said, we find that particular adipocytes are those cell types which have most of these changes. And from there, we then moved on and said, okay, this is obviously very challenging now to study in much more detail these things in humans, particularly because we need now cell type specific information. We want to maybe do more interventions. We may want to reduce potential genetic effects, which while we control them as much as possible in humans, we obviously still a diverse population. And so we moved to look in the same thing in the mice. And so we set up a mouse cohort where we fed the mice, put them on an obesogenic diet, they gained weight, and we more or less in the initial phase repeated exactly the same experiments with the humans. So they gained weight, they lost weight. And here again, we find very similarly a number of genes change during the weight gain phase, and many of those actually are deregulated even after weight loss. Metabolically, equally, these mice normalize, and so are, again, I would say, if you look at most of the metabolic parameters, almost indistinguishable to the wild type in non-obese litter mates. And once we had seen that, we obviously asked what are potential contributions to that. And that's where we then took advantage that there's also genetic models which allows us to label the adipocytes. And so we could now, from the same cohort, label adipocytes before we put them on this high-fat diet, and then at the end isolate these adipocytes which have been experienced either the whole weight gain period or the weight loss period, and then ask, are there in addition to these transcriptional changes also epigenetic changes? And that's indeed what we find. So if we now did this first a multifactorial analysis, it really allows us to combine all of these data and look, we clearly find that those animals which have never experienced obesity, have never been fed this high-fat diet, are quite distinct to those which have experienced that. And interestingly, even those which are back to this normal state, so again, fed a normal diet, have lost their weight, they still are quite distinct to those who have never experienced that. And so we find then that there is many promoter regions which have gained or lost K27 trimethylation and K4 trimethylation, through this key active or repressive modification at the promoters, and they are maintained similar to the transcriptional signatures. We equally find that many of those actually overlap that. So really many genes where we see the transcription effects, they also have these epigenetic effects. Equally find similar changes at the histone, at the enhancer landscape. And so I think overall this showed there is an epigenetic memory, and we call that memory built up during weight gain, and that is not completely erased after weight loss. And finally, we always ask, is this functional? Because at this point, it was a nice correlation and indicating that maybe there are some effects. And so what we then did is we re-exposed these animals to a high-fat diet and find that this re-exposure of those who have prior been obese are much faster regain weight. Also, their adipocytes are different. They're much faster to take up glucose, to take up fat. And equally, if we now look at the enhancer landscape, we can even make some predictions of the later transcription response after weight regain and find again their correlations. And so overall, we believe that this really indicates that in the adipocytes, a memory is built up at the epigenetic level, at the transcription level, which then really has a function implication. And so this may contribute to this faster reweight gain that we see in the mice, but also in humans. Obviously, a small caveat I should add that the data we have at this point is largely correlations. So we have not yet been able to really do the intervention at the epigenetic level of modifying it and that kind of erasing that. But that is something obviously at this point with not only one or two genes being or regions affected, but by as many thousands being affected is still quite challenging. But still something we hope to get a bit more control and a bit more insights over in the next few years.
And I guess also working on therapeutic interventions, right? To reverse it, maybe.
Exactly. That's actually now the big goal now that we're starting in the lab. So this idea, can we start reversing epigenetic states? So a little bit moving forward on that, I think in many therapeutic ways, if we can start modulating the epigenome, I think that would be fantastic. And that's really the goal now, not at large, but a good part of the group now really trying to work out epigenome modifying strategies, targeted ones. Is that possible? And if you can move forward with that, I think that would be fantastic in the context of the adipose tissue. But also in many other regions, we find also changes in the liver. We are now quite interested also in the brain or the pancreas. So I believe in many of these, the potential is there that long-term environmental nutritional changes affects the epigenome and may then be contributing to the disease state that we find.
That's also very interesting because not only does this play a role in obesity, but also if you have been fit and have been working out or have been at a certain stage, then you may be injured and then you get back to working out again. That's the same effect, right? You also can get back into shape much quicker once you have been in shape. So there seems to be some similar mechanisms going on.
I mean, these could be at probably many levels. I would not say epigenome is everything here. I think that would be slightly overstating that. And so there are also in the adipose tissue, we see some more changes at the extracellular matrix and many other sites that probably are part of that. But clearly at the function level, the epigenome probably plays a key role. And so we have also with a Spanish group obtained samples from humans which have been exercised at various levels. And at the moment, starting to look a bit more into that. And maybe there's also really effects of high intensity training, no training. And I think that's, we're quite interested in all of this modulation that are existing there.
So I've been talking about what you want to do in the future for a bit, but maybe we can put it together in one answer again. Imagine you are due to submitting a grant proposal tomorrow. What would you have written into that?
which we actually did recently.
Yeah, of course.
And so actually one of the big grants we at the moment hoping to move forward with is on epigenetic editing. Really targeted modulation, not only of one modification, but kind of a combinatorial effect of several modifications. And I think what we want to do is a little bit rethink of the classical epigenetic editing with the Cas9 system using a slightly different approach, and then hopefully reinstate healthier epigenetic states. I think that's one of the key goals we have, because I think this would then have application in metabolic diseases. It could have in cancer context, basically in all kind of disease states, and ultimately maybe in the whole more aging related context, which I think is a very exciting area to further explore.
So for the last 45 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I think maybe one thing is many of the things that I've done, and I think also with my group now, and also with colleagues before in Wolf's lab and in other places, is it really we followed some exciting findings. And not all of these findings have been like we said, okay, this has to be the case. That's what we really want to do. It's more like we try to look for it. And many times I think the exciting part of that was having at the various institutions I was in with the various groups and members and also the supervisors, really the opportunity to follow exciting leads rather than having to fulfill a specific mission. And I think that was really exciting, and that's something I really enjoyed. And also I hope for the people in my lab somewhat I can give the opportunity to really find something, get excited about that and move forward with that. And so this is really kind of this biomedical space, I think, has so many exciting areas that would be fantastic for the future also.
To finish, what is your personal definition of epigenetics?
I think I'm quite traditional. I think this is heritable changes affecting cell function without affecting the genetic code. I think that's kind of the minimal element of that. And I also don't think we have to limit that to specific modifications, to specific things. I mean, I think I'm very inclusive in that respect.
So thank you, Ferdinand, for your time and for being on the show.
Thanks a lot. It was great.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.